Okay, now we go into the discussion of fluorescent yield, or usually we call as quantum yield. Sometimes when you read journal or textbook, you will see that it is also known as quantum efficiency. So what is quantum yield? So it is a measure of how many photons, okay, packet of energy, end up in the fluorescence or phosphorescence channel compared to the number of photons that were lost into the non-radiative pathway. Apa itu non-radiative pathway? So all those internal conversion, external conversion, intersystem crossing, dissociation and pre-dissociation. So these non-radiative pathways are detrimental to the fluorescent because it can be due to the several factors that we discussed before. Okay, for example, the structure, the solvent, the temperature, the pH. Okay, so we want to know how much of the fluorescent as compared to all these sort of like culprits. Okay, so this is the symbol. Okay, if it's F is fluorescent, if it's P is phosphorescent. So how do we get fluorescent intensity? Okay, fluorescent intensity is related to the molecular extinction coefficient and directly proportional to the intensity of the initial radiation. Radiation yang awal-awal masuk, okay? So this is the incident radiation, and we have the quantum yield, okay, will make up the intensity of the fluorescent, macam absorption, okay, in UV visible radiation. But we have also the same parameter, concentration, okay, and also we have path length, and then we have the factor with the epsilon. Okay, usually for intensity for the fluorescent can be easily calculated when we have quantum yield over here. What you can do is that you can have a look on the quantum yield for each fluorescing compound in textbook, whereby it has been tabulated for you to use in any application if you are analyzing with fluorescence spectroscopy method. So what makes a good fluorophore a good fluorophore? Okay, it must have good spectral properties, okay, because of the structure. Okay, strong absorber of the light. What light? Okay, strong absorber of the radiation given. What kind of radiation? It can be from UV, but can also be in the region of radiation. In the region of visible. Okay, but most of the time is UV. Okay, we want to have high quantum yield for fluorescent, but we don't want high quantum yield for all those non-radiative effect. Okay, so we want to have low quantum yield for loss. Loss ni kena kurang, photo destruction kena kurang. Okay, so that fluorescent will be high. Okay, a good fluorophore usually is a small molecule easily attached to biomolecule or any biospecies. This is as a fluorescent tracer. Remember in the first few video in fluorescent, I mentioned about biochemists or medicinal chemists use fluorescent compound so that they can see fluorescing compound under the microscope. Okay, let us see on the instrumental part of fluorometer. Basically, it is almost the same with ultraviolet visible spectroscopy, except it has two set of filters. What kind of filters and why there are two filters? Remember, okay, the spectrum. Remember the spectrum? Okay, we have one spectrum of absorption or excitation, and then we have one spectrum for emission, what we call as lambda emission and lambda excitation. Absorption lah ni Okay, because of that we need two filters to filter out one excited one and one emitted. That's why in fluorescence spectroscopy, I really like it because I have two data. I can have two data. I know what is the absorption lambda, and I know what is the emission lambda. So you have two data as compared to UV visible spectroscopy. So let us look on how the instrumentation in more better diagram. Okay, so we have radiation over here. Okay, we have excitation monochromator, excitation laser. It is also, it can be using laser as well. And then you see that the color is different over here. This is more towards a faint purple, and then it goes into violet and blue because the lambda is shifting to the longer part, right? So here we have at shorter wavelength, sini excited lambda, and then we have emitted lambda over here. Okay, longer wavelength, color pun naik, intensity increase. So we have emission lambda. Why you see the monochromator is situated 90 degree from each other? Why do you think? Okay, because we don't want to get a stray light when you don't put in 90 degree angle. Okay, imagine if you have emission monochromator sini. Okay, whatever has been absorbed, kita kan ada also the transmittant ada extra kan. So rather than collecting the emitted radiation, you are collecting all those transmitted, the one that is not fluorescing. So your filter over here will capture this rather than this emitted one. So it is important for you to position 90 degree to each other. Sini. Okay, rather than here. Sini tak boleh, it's 90 degree. 91 degree also cannot. Okay, it's usually hard to get like this. So this is the example of a spectrophlorometer. We have one spectrophlorometer, but it's not been used largely. Therefore, the maintenance is not up to the standard lah. Okay. So this is the source of radiation. Okay, if you have tungsten lamp ni, it can give you UV radiation. Okay, if you have xenon arc lamp, it can give you UV until visible and also near IR. Okay, and you can use mercury lamp, and also you can use laser, but laser is mostly in the mid UV range up to visible. Okay, the same thing with a UV visible spectroscopy. If you use only glass filter, you will call it as fluorometer. When you use monochromator, we have slit, prism or grating, and also slit again. So you have two set of this, you call it as spectrofluorometer. Okay. So this is the spectrum that has been shown to you before many times already. The cell holder is the same like UV visible spectroscopy, mostly better use quartz. Okay. Detector same. Charge couple detector enable the collection of multiple wavelength at one time. Okay, so this is the best thing about CCD. It collect a lot of radiation at one time. Okay, you see that the fluorescence the fluorescence spectra, okay, increase from very diluted concentration up to more concentrated concentration. Okay, but as I mentioned before, it is linear only to a certain concentration. The same thing, it will reach plateau or even go down because of the collision. Kan, collision frequency meningkat. Okay, and then the conversion is also increased. Therefore, you decrease the fluorescence. Okay, So this is increasing up to certain range only. That's why it's very important for you to do graph. Okay, spectra over here, the same thing. Okay, we will do active learning on extracting information from a journal article. Okay, this is a journal article, or rather we call it as technical article. Okay, given by, I think this is the brand who produce a spectrophlorometer in our department. Okay, so I will give you the paper on determination of quinine in tonic water using fluorescence spectroscopy. Quinine ni asalnya is a ubat anti malaria. Okay, it's also good to be used as flavor. Okay, kat sini nanti okay, we will see the journal article, and it is using think pair share, means that pair with any of your friend, okay, as long as it is pair, and then you extract information on the experimental and results of fluorescence. Okay, fluorescence fluorescence detector.